To IGCSE Biology, Chapter 21 Biotechnology and Genetic Modification. Why are bacteria useful in biotechnology?
Because bacteria reproduce rapidly, can make complex molecules like insulin, enzymes, have plasmids which are used in genetic engineering. Bacteria share the same genetic code as humans. Cheap and easy to grow. No major ethical concerns.
Suggest advantages of producing products using genetically engineered bacteria.
Reliable and constant supply. Large quantities can be produced quickly. No dependence on blood donors. Lower risk of disease transmission.
Explain why nutrients are sterilized before being added to the fermenter.
Nutrients are sterilized to kill unwanted microorganisms, prevent contamination, stop competition for nutrients.
Why must a fermenter have a gas outlet pipe?
Carbon dioxide is produced during respiration. If gas is not released, pressure would build up. The fermenter could explode.
Name the microorganism that produces penicillin.
Penicillium, a fungus.
List the nutrients needed in a fermenter to produce penicillin. Glucose, ammonia, amino acids, mineral salts, water. Name the gas that makes bread rise. Carbon dioxide. It is produced during anaerobic respiration by yeast. Outline steps in penicillin production.
The fungus is grown in a fermenter. The fungus reproduces rapidly. When nutrients become limited, growth slows. Penicillin is produced as a byproduct in the stationary phase. The penicillin is filtered and purified.
Explain why different temperatures are used in bread making.
At around 35 degrees Celsius, yeast enzymes work best. Respiration occurs. Dough rises. At around 200 degrees Celsius, yeast is killed. Enzymes are denatured. Bread is cooked.
Explain why pectinase is used in fruit juice production.
Pectinase breaks down pectin in cell walls, increases the amount of juice produced, makes the juice clearer, speeds up the process.
Why are enzymes trapped in beads when making lactose-free milk?
Because they can be reused. It is cheaper. No enzyme remains in the milk. Milk does not need purification.
Why are biological washing powders effective at lower temperatures?
Biological washing powders contain enzymes. These enzymes break down proteins and fats. Work best at 30 to 40 degrees Celsius. Remove stains at lower temperatures.
State 3 conditions controlled in a fermenter. Temperature, pH, oxygen level, carbon dioxide level, nutrient supply. List the uses of anaerobic respiration in yeast. Bread making, brewing, beer production, wine production, biofuel production. Explain the mechanism of action of biological washing powder.
Biological washing powder contains enzymes. Proteases break down proteins into amino acids. Lipases break down fats into fatty acids and glycerol. These smaller molecules are soluble in water. This allows stains to be washed away easily.
Why stirring is essential in a fermenter?
Stirring ensures even distribution of microorganisms and nutrients. Oxygen is mixed properly throughout the fermenter. Nutrients remain in solution. Cells do not settle at the bottom. Growth is not limited in one area.
How to identify antibiotic resistance?
If bacteria are resistant to an antibiotic, there is no clear zone around the antibiotic disc. The clear zone of inhibition, is very small. Bacteria grow right up to the disc.
List the steps in genetic modification.
Identify and isolate the required gene using restriction enzymes. Cut a bacterial plasmid using the same restriction enzyme. Insert the gene into the plasmid. Join the DNA using DNA ligase. Insert the modified plasmid into a bacterium. The bacterium reproduces and produces the desired protein.
Give examples of how genetic engineering is used in agriculture and medicine.
Insert human genes into bacteria to produce insulin. Make crops resistant to herbicides. Make crops resistant to insect pests. Improve nutritional value of crops.
Explain why the temperature inside a fermenter must be carefully controlled.
Enzymes work best at an optimum temperature. High temperatures can denature enzymes. Low temperatures slow down respiration and growth.
Define genetic engineering. Genetic engineering is the transfer of a gene from one organism to another to produce a desired characteristic.
Explain the role of restriction enzymes in genetic modification. Cut DNA at specific base sequences. Cut open the plasmid.
State advantages of genetically modified crops. Higher yield, resistant to pests, resistant to herbicides, improved nutritional value, reduced need for pesticides.
Explain why plasmids are used as vectors in genetic modification. Can carry foreign genes, replicate inside bacteria, transfer the desired gene into bacteria.
State one difference between genetic engineering and selective breeding.
Genetic engineering transfers specific genes directly. Selective breeding selects parents with desirable characteristics over many generations.